Next, we're going to start talking about the nucleus of the cell. Now, the nucleus is, some people consider it an organelle, but it's a very, very specialized organelle, so I consider it also to be a distinct part of the cell. Again, plasma membrane, nucleus, cytoplasm are the three parts of the cell. So the nucleus, we've already got a phospholipid bilayer, but the nucleus is going to take a membrane-bound organelle title to a new level. We're going to take that phospholipid bilayer and we're going to fold it into two layers of phospholipid bilayer. And so we're going to have an inner membrane and an outer membrane. This is really going to separate that genetic material from the rest of the cell. The only way that things can come and go from that nuclear environment is through a nuclear pore. So the nucleus has its own pores. Now, inside of the nucleus, we do have the genetic material, which could be in the form of chromosomes or chromatin. Chromosomes are wound, chromatin is loose. And we also have a structure known as the nucleolus, which is the site of ribosome assembly. That's where you are making ribosomes. We'll talk about ribosomes more in just a minute. Now, a chromosome is a form of genetic material where the DNA has been wound. So we start with a long, thin strand of DNA. Very, very, very long, very, very, very thin. In fact, every single chromosome in your body, and you've got, let's see, 46 in every single cell of your body, but you take just one chromosome, you stretch it out 6 feet of DNA. Literally 6 feet, 2 meters-ish of genetic material in one chromosome in one cell. It's a ridiculous amount of information that we are carrying around with us. So in order to get that 6 feet of information into a microscopic chromosome that can be seen with a light microscope when it's in the form of chromosome, we are going to take that long strand and we are going to twist it like an old school telephone wire. Maybe you're old enough to have had one of those coiled telephone wires. Maybe not. One day I'm not going to be able to teach this way anymore. I'm going to be really sad. I'm old enough to have had the coiled telephone wires. So we're going to start coiling it. And the first thing we coil this strand of DNA around is a protein known as a histone. So we wrap the DNA around histones, and then you get a structure that looks pretty much like that telephone wire. If you are old enough to have played with those telephone wires, if you ever got the phone twisted around and those coils got a little too tight, did you ever notice that they would like wrap around themselves and you'd end up with something that looks like that? That's what you're going to do at your next level. So you're going to wrap around histones and you're going to have this tightly coiled thing. That coil, you are going to continue to coil until it wraps around itself. And then you're going to coil that coil, and you're going to coil that coil, that coil, that coil it. And we're going to keep coiling the coils, many orders of magnitude of coil, until we've coiled 6 feet of information into something that can barely be seen with a light microscope. And every time we go from loose chromatin, just the DNA, like loose like spaghetti is chromatin, every time we coil that up into a chromosome. It's actually part of mitosis, to coil it up into chromosome. So that's why you have two forms of DNA. When it's loose as chromatin, you can read the genetic material. You can actually use that for transcription, which we'll get to in just a bit. When it's bound up into a chromosome, it's tightly packed, nothing you can do. It's there to be transported. I think of genetic material as like a blueprint. It's a set of instructions. So imagine this is a blueprint. When it's chromatin, the blueprint is rolled out on your drawing board. You can use it to build a chair or a house or whatever it is you need a blueprint for. But when you coil it, it's like rolling that blueprint up, and now it's easier to transport to another place. So now I'm getting to the idea of the factory metaphor. I have an overextended metaphor where the cell is a factory. So if the cell is a factory, genetic material is blueprints, the nucleus is the manager. Let's pretend that managers are highly effective. This is a very effective manager that is directing the manufacture of something. And we'll get to what that something is a little later. got all that. Got all that. So that was the nucleus. We'll move on to the endoplasmic reticulum. We can talk about protein synthesis in just a bit. And in fact, this is the start of protein synthesis. You have two forms of endoplasmic reticulum. We're on to cytoplasm. We're talking about our first membrane-bound organelle in the cytoplasm. The endoplasmic reticulum has two forms, rough and smooth, RER and SER. Rough endoplasmic reticulum is associated with ribosomes. Ribosomes, remember, came from the nucleolus. They are made out of protein. They're also made out of nucleotides. You do have the same building blocks as genetic information have to structurally be part of this ribosome. And when we talk about transcription and translation, we'll talk about why that is. So the ribosome associates with the endoplasmic reticulum, and that allows us to synthesize proteins. So we take amino acids, we bond them together into polypeptide chains. The polypeptide chains fold into a functioning protein. So that's your rough endoplasmic reticulum synthesizing a protein. This is the ribosome. It came from the nucleolus. It has a large unit and a small subunit. Ribosomes can be membrane-bound on the rough endoplasmic reticulum. You may also have ribosomes elsewhere in the cell. You can have free ribosomes as well. The difference between free and bound is actually where the proteins end up that they synthesize. So there is actually a difference between protein synthesized by RER and protein synthesized by free ribosomes. It's not the most important thing in biological sciences, at least according to me, but it's still cool. So again, rough endoplasmic reticulum is helping with protein manufacture. Smooth endoplasmic reticulum does not associate with ribosomes. We're still going to use it to build stuff. We're going to use it for our other macromolecules of life, such as lipids, and it's not on here, but also carbohydrates, and it's going to help with detoxification as well. It's not on here. I try to stick with things that are actually in my PowerPoints, but I think it's important enough to recognize that there are 4 macromolecules of life, and this is only addressing 2. The nucleus addresses the nucleotides, but we got to have our good friend carbohydrates in here somewhere, don't we? The endoplasmic reticulum is located close to the nucleus. In fact, some of this membrane is derived from the nuclear membrane. It's continuous. What we've done, again, this was a 3-dimensional structure, and we just bisected it. So this would continue in the opposite direction. These would all continue in the opposite direction. All of these little cisterns, all of these cavities and spaces would continue and be continuous structures. We can see with some of our microscopy techniques how rough endoplasmic reticulum is going to look, and we can see and visualize individual ribosomes on it. Again, this is just about, this is going to tell us about some transcription and translation, which we're really going to address a little bit more later. So I'll come back to that concept. Farther out from the nucleus, again, we went nucleus, RER, SER. Now we have a Golgi apparatus. Now, I mentioned a factory metaphor. Endoplasmic reticulum, for me, is like the actual assembly line of your factory. You made things. You made proteins, you made lipids, and again, I'm mentioning that you're detoxifying and you're making carbs as well. So you made stuff with the endoplasmic reticulum. The Golgi apparatus in our factory metaphor is packing and shipping. You've made something. You gotta get it to where it belongs, where it needs to go. So some of the things that you synthesize in a cell are going to end up outside of the cell. Some of them are going to be incorporated into the cell. Maybe they'll be incorporated into the plasma membrane. No matter where it's going, you've got to get it there. And the Golgi apparatus is going to get it there. So you do have these stacks called cisterns I think of it like a stack of pancakes. You got a big old stack of pancakes going on here. They've just bisected that stack of pancakes so that you can look into the cisterns. It is directional, so you receive things that were synthesized by the endoplasmic reticulum. They go through these series of cisterns. The inner face receives it, the outer face sends it. The outer face packs things into vesicles, which again, just a sphere of plasma membrane that contains something important to us. And that's it. That's your Golgi apparatus. It does have a little bit left of refining. So let's say you synthesized a protein with your rough endoplasmic reticulum and you sent that to your Golgi apparatus. It can make any final adjustments. Any final folds in that protein can happen in the Golgi apparatus. So maybe your packing and shipping department of your factory is responsible for polishing up that chair before it gets packed off to the local furniture store. Yeah. So again, you do have different kinds of vesicles. You have vesicles that just contain secretory molecules, maybe the thing that you synthesized during your synthesis on your ribosomes was a neurotransmitter, and that's going to undergo exocytosis. That's possible, or exocytosis rather. There's some variations of vesicles. Some of them are lysosomes, which means that they contain digestive enzymes. So if something has engulfed via endocytosis, we can have it meet up with a lysosome, which contains digestive enzymes. They fuse, and the digestive enzymes in the lysosome, chomp, 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 eat away at the thing that was engulfed. And that is actually phagocytosis. Or maybe the lysosome is just going to engulf waste products of the cell itself. Peroxisomes help with detoxification. Especially if things like, still contain some things like catalase enzyme. I don't know that we're going to have a lab for catalase. I'll look for one. But catalase is an important enzyme that breaks down H2O2, hydrogen peroxide, which is the stuff in the brown bottle in the medicine cabinet. It is also a byproduct of some metabolic activities. So your cells are making hydrogen peroxide right now, and it is cytotoxic. So we really don't want hydrogen peroxide in our cells. So we do have dedicated structures in our cells for breaking down H2O2 into something that we can get rid of. And your peroxisomes have a big role to play in that. Oof, okay. So mitochondria, if we were in class, if this was a synchronous class, this would be the moment where I find out who my internet people are. Hi, internet people. What does the mitochondria do? That's right, it's the powerhouse of the cell. Hello, internet friend. I too am of the internet. Okay, so the joke is that the only thing you learned in high school is that mitochondria is the powerhouse of the cell. They didn't teach you how to do taxes or exist as a good citizen of the country. What they taught you was the mitochondria was the powerhouse of the cell. How dare they? Again, physiologically, this is pretty important to us, that we're going to take oxygen and glucose, and we're going to turn it into ATP, heat, and carbon dioxide. We will have our own lecture just for cellular respiration. We will go into that separately. But this is where the aerobic parts of cellular respiration take place. This is how we are able to generate a lot of ATP for the cell. I like how it's just over my head here. ATP. That's great. It is how we're going to power all of our pumps, how we're going to power all of our cellular activities that are important to keeping us alive. So if I ask like a question about what does ATP do, or sorry, what does the mitochondria do, I don't really like for it to be as simple as it's the powerhouse of the cell. I think that's too colloquial, and we are scientists now, so we do have to be more scientific than that. The mitochondria participates in the aerobic portion of cellular respiration to provide ATP for cellular energy. It does have an inner and an outer membrane. The inner membrane is invaginated into cristae. There's a matrix. There you go. Oopsie. Okay, non-membrane bound organelles. All of those were based on membranes. So we're finally getting to ones that are not based on membranes. These are predominantly made out of protein. Cytoskeleton is definitely mostly made out of protein as a structure. Most of the things your cells can build are proteins. The functional parts of cells, those are all going to be proteins. Stuff that has a job to do, that's a protein. So the cytoskeleton supports the cell. Variations on cytoskeleton are going to do some other things as well. They are also going to move the cell, either movement of the cell itself or movement along the surface of the cell. So something outside of the cell is going to move when parts of cytoskeleton move. And there's also going to be variations of cytoskeleton that guide mitosis. So structure and support, movement, mitosis. Those are the things that cytoskeleton can do. We have variations on cytoskeleton, like microtubules, microfilaments. They're made out of slightly different proteins and slightly different arrangements. They have different sizes. So one organelle associated with specifically microtubules are cilia and flagella. Now, cilia and flagella are both the things responsible for movement. There's only one type of cell in all of the human systems, plural, that has a flagella, and that is in the biologically male reproductive system. The sperm is the only human cell with a flagella. Now, many cells in the human body have cilia, so the surface of the cell will contain many cilia capable of moving, and they usually move in concert too. Whether it's a flagellum, which is singular, or cilia, which there are always going to be many of in the human system, we are talking about microtubules in a specific arrangement. So we're going to have a microtubule pair. We're going to have 9 microtubule pairs. And right in the middle, we're going to have a little microtubule doublet. And functionally what's happening here is where these pairs are, there's proteins that are kind of linking them together. So each of these pairs is going to essentially rub against the other, and collectively, when this 9+2 microtubule arrangement is operating, those individuals rubbing against each other is going to collectively cause the movement of either the cilia or flagella. So that 9+2 arrangement works in both the cilia and flagella to cause movement. We talked about phagocytosis already. Phagocytosis is when we use exocytosis to engulf something. Maybe this is for immune support. Maybe this is a bacterium that we've engulfed here. And then we take this vesicle that is filled with the bacterium, we'll call it a phagosome. We will merge it with a lysosome, which contains digestive enzymes, and then those enzymes can break down that bacterium. And hopefully this is something that we can get rid of. There are things in the body that we can't get rid of, and they hang around as little dusty particles that clog up our cells and stay permanently. There's scenarios where that happens. Sometimes it's something that we can excrete and ultimately can leave our bodies. It depends on what was broken down. So just to break down, wrap up this lecture on cells, metabolism. What is metabolism? Metabolism is when we break down macromolecules of life. We go from large to small. We get them all the way down to that building block level of macromolecule, that amino acid or that glucose molecule or that fatty acid little lump that you get. That would be catabolism. Breaking something from large to small is catabolism. Breaking it down is catabolism. When we take the building blocks of life and construct it into something new for the cell, that's called anabolism. So catabolism, breaking down, plus anabolism, breaking up, is metabolism. So I'm going to go ahead And while I'm recording, just take this note for you, or have you take this note along with me. Catabolism is breaking down. Anabolism is building up. So we're always going to have a lot of enzymes to help us with these processes. We're going to have enzymes to catabolize. We're going to have enzymes to anabolize. An enzyme, again, being a protein with a job to do. So anabolism is important in the cell because we're going to take all of the building blocks of life and we're going to build them into components for cells. We're going to build them into cytoskeleton. We're going to build them into ribosomes. We're going to build them into centrioles, which I think I forgot to mention. Centrioles guide mitosis. This is going to help form the mitotic spindle. So we'll talk about that more with our mitosis lecture. So you wouldn't have cells if you didn't have anabolism. You have to be able to build stuff up. And of course our factory, our cells are responsible for doing that. So metabolism is a cellular activity. Metabolism happens in cells. Now, one specific metabolic pathway is cellular respiration. We're going to catabolize glucose. We're going to utilize oxygen. And that's going to lead to the synthesis of ATP with a byproduct of carbon dioxide and water. That ATP can then be used for cellular work. That ATP can be used for anabolism because when we go from something simple to something complex, that tends to require energy. A lot of different processes in cells are going to require energy. We've seen one so far, the energy of pumping things against gradients. There's also the energy of muscle contraction, neurons firing, the energy required to transport something from one place to another. That's all going to require ATP. Those are all going to be very energy-hungry activities. That wraps up the cell component. We do have several other cellular mechanisms to discuss. Some of them aren't even from Chapter 3, but they're all important to address right now.